Yours.
Okay, hello, good afternoon. I'm also very happy to be here with all the nice researchers and all the presentations that preceded my humble short lecture. I'm very interested in virology. That is my area of study. That is why I'm very happy to be talking about what really drives me and what really keeps me interested in doing science. So we'll be talking about antiviral properties, potential antiviral properties of natural products. And also in my field of science, we were also testing some semi-synthetic or synthetic compounds. However, still natural products are definitely more interesting to me. They are more versatile. They have more interesting properties that can be tested using various types of tests. And definitely, this is the best research material, at least for me. Of course, we all know that natural products are extremely diverse. They can be obtained from any living organisms, from plants, from animals, also from microorganisms. And in recent years, we have a huge development of studies on the marine organisms, which are also a source of enormous source of bioactive molecules. The natural products gave us a lot of different drugs, a lot of different bioactive molecules, including drugs used for heart failure, like digitalis biocompounds. We also have tubocurarine, we have alkaloids, various types of alkaloids. For example, morphine, which is used as a painkiller, and also various other opioids. We have obviously cytotoxic drugs derived from natural products. Most of them are actually derived from natural products, like vinca alkaloids. We also have Taxus baccata, which is a source of Taxol. And finally, the natural products may also be a valuable source for antimicrobial compounds. However, when we talk about antimicrobial activity of natural products, we are usually thinking about antibacterial drugs like penicillin and all the other antibiotics we have. We usually do not focus on antivirals, and to be honest, most of the people and also the scientists think that those antivirals are only synthetic drugs. There are no natural products with antiviral activity, which is not necessarily correct, because when the whole antiviral therapy began, the first drugs which were introduced were actually synthetic drugs, but they usually had very high toxicity and thus limited application. The big change came in 1950s, when the Bergman and his co-workers found a remarkable substances in a Caribbean sponge, which was Tessitaia crypta, and it occurred to them that those molecules were not found before in any other living organism, and these were unique because they were nucleosides without a normal sugar like ribose, but instead of it, they had arabinose sugar. This was very interesting. As I said, it was very unique as well. And this led to introduction of the first two marine drugs, which were actually used in the treatment of human diseases. And these were vidarabine and cytarabine, ARA-A and ARA-C. These drugs were used in case of herpes infections, like ARA-A, or they had some nice cytotoxic properties with application in leukemias or non-Hodgkin's lymphoma. However, this does not end here, because they were further developed. A lot of different semi-synthetic modifications were performed to obtain structural analogs, and because of this, we have obtained new antivirals, which are actually nowadays we use, like acyclovir, which is icycloguanosine, or azidothymidine. So, to be honest, acyclovir is not a natural product. However, it was obtained thanks to the study of natural product, because it all started with the studies on Thesitaya crypta. Since then, we had a huge development in antiviral drugs. We had, especially in recent years, starting from 2013, approval of the first direct-acting antivirals like sofosbuvir, and this completely revolutionized the treatment of hepatitis C, for example. And nowadays, the efficiency of the treatment is close to 95%. So basically, this was a huge advancement. How does it look in case of the origin of those molecules? This is a study of only those drugs which were approved beginning from 1981 to 2006. The study was published in 2007. And as you can see here, the authors listed the origin of the molecule. Of course, some of them are biological substances like interference. Others are synthetic. However, most of them are actually based on natural products. They are not natural products present in plants or other organisms. However, the pharmacophores were very commonly taken from a natural product and then developed further on to produce antiviral substances. And if we sum this up, we will see that 44% of the antivirals approved between this period were actually either natural products or semi-synthetic natural products, the analogs of them, or synthetic compounds based on those natural product pharmacophores, which is actually very interesting observation. Well, in the previous session, we had a nice presentation about betulinic acid. That's why during the pause we had between the lectures, I've added 2 more slides to my presentation because it gave me an inspiration to a little bit broaden my talk. And I would like to talk about the betulinic acid as potential anti-HIV drug, because such tests were performed, and actually it gave very nice results in vitro. So nice results that they have been further developed in clinical trials. However, when it came to clinical trials, there were significant issues with resistance that emerged during the clinical trials. That's why eventually those trials were suspended. However, it should be mentioned that betulinic acid proved to possess a very unique activity as a maturation inhibitor of the HIV virus. Well, we have a lot of different antivirals to be used in the treatment of HIV infections in the highly active antiretroviral treatment. However, they all have different than betulinic acid mechanism of action. We have inhibitors of the reverse transcriptase. We have inhibitors of integrase or protease. But this is, in case of betulinic acid, a completely novel and previously untested kind of activity. So this was quite interesting, which led to further research and introduction of the first and the second generation of derivatives. Both of them were tested and proved to be extremely active in vitro. They were developed into clinical trials. The first one in 2007, it was tested in phase 2B clinical trials. However, then it was purchased by Myriad Genetics, and then Myriad Genetics soon after changed their portfolio, and they have focused more on anti-cancer drug research. That's why they've halted the further development of this molecule. The second one was also tested, was also in phase 2B clinical trials. However, they were discontinued because of certain adverse effects, and once again, the development of drug resistance. However, this kind of activity as a maturation inhibitor is very interesting. A major problem with this is the polymorphism of GAG protein, which is actually responsible for the fast emergence of resistance. The second example I would like to mention, even more interesting, is the calanolid A. This is an example how certain great discoveries are made by chance, just by accident. Well, there was an expedition to Borneo in 1987, which was sponsored by the National Cancer Institute. And obviously, the major goal of National Cancer Institute is to look for cytotoxic drugs, to look for anti-cancer drugs. And they found in a forest in Borneo several species of Calophyllum. They have taken the samples, they came back to the United States, they've tested the anti-cancer potential, and they found no activity. That is why we would think that the story ends here, but we forget about the fact that inside the NCI, there is also a lab focusing on anti-HIV studies. And they did the assays against the HIV virus, and they found that a single species called Calophyllum lonigerum had the activity against the HIV virus. The problem was that the sample was very little. They could not perform isolation or further analysis, so they had to go back to Borneo and collect some more of the plant. It was also a problem because this plant was only found on Borneo. So they went back to Borneo, and the forest where this plant was normally found was not present anymore. It was destroyed. And they could not find a single plant left. They've searched, they've asked people, and no one knew about this plant. Fortunately, they found just a few of the specimens of this particular species in a botanic garden in Singapore. So they could continue the studies, and they found that these are the calonolid A was responsible, and it had a remarkable activity as a reverse non-nucleoside reverse transcriptase inhibitors, but it was also different from all the other reverse transcriptase inhibitors because it bound to two distinct sites on the enzyme, which is interesting because other drugs, they bind with only one of them, which made it so remarkable. However, because of side effects and delayed clinical effect, the studies on this particular molecule were discontinued. However, it gave an idea to other scientists which further developed it. A lot of analogs were synthesized. This is from 2010 and 2012. And then the laboratory run by Professor Ian Balzarini managed to think of alternative way of utilizing calonolid A. They took the pharmacophore and they fused it with, they tried to fuse it with various molecules, and they came up with a completely new scaffold with a potential anti-HIV activity. And Professor Ian Balzarini is one of the most famous virologists who was working about on several different reverse transcriptase inhibitors, which are currently on the market, and they are used actually to treat people. So maybe in future, if this goes further, we will have some more new potential non-nucleoside inhibitors of the reverse transcriptase. So that was like a little bit of introduction to my talk, inspired by previous talks I've heard today. When we talk about viruses, we have to remember that these are obliged intracellular parasites, meaning that they can replicate only inside living cells. By utilizing their biochemical and molecular machinery, they're capable of making more copies of themselves, which makes studies of antiviral activity completely different from the studies of antimicrobial potential against bacteria or fungi, because here we have to take into account the use of living cells, human or animal cells, which are capable of sustaining the replication of the particular virus. The antiviral drugs or any other molecules with antiviral activity must target specific steps in the virus replication cycle. This is an example, a very simple example of the replication cycle of HIV virus. And as you can see right here, there are certain steps like reverse transcription, like integration, or the production of structural proteins by the protease, which may be actually influenced by antiviral drugs. What is very important, that In case of antiviral drugs, the molecule we are testing has to be able to penetrate into the cell and interact with these specific steps of viral replication without affecting the cellular metabolism, because we don't want the cells in a human body to be affected, normal cells to be affected by the drug. Though these drugs have to be safe first for the cell and then be able to suppress the replication of the virus. The viruses we are studying in our laboratory, most of them are causing cytocidal infections, meaning that their effect on physiology, biochemistry, or other biological effects leads to morphologic changes, and basically those cells die during the infection with a virus. Of course, some viruses differ from this because they are causing different kinds of persistent infections. However, today we will not be focusing on these types of viruses because the methodology of study will be a little bit different in case of those persistent infections. Before we start testing for any kind of antiviral activity, the first question we have to ask ourselves is what we are looking for. Because either we are testing virucidal activity, meaning that the ability of molecules to destroy viral particles outside human body, or are we testing for antiviral properties, so the influence of molecules on the viral replication cycle, which happens inside human body. So that is the first decision. Actually, in our laboratory, we are not doing virucidal studies usually, with some exceptions. We are focusing on antiviral studies. And in order to do this, we must select the virus we want to test, we want to use for the assessment, then select the cell line which will be capable of being infected successfully with this particular virus. Then we have to culture the virus to high enough quantity to be able to use it in further experiment, and then we can attempt the actual testing, which is always preceded by the evaluation of the cytotoxicity. Only after knowing the cytotoxicity, we can go further to antiviral activity. The replication of the virus on a cell culture results in different kinds of cytopathic effects in case of cytocidal infection. And usually this is how our viruses like herpes viruses or Coxsackie viruses behave, or adenoviruses. They will be inducing different kinds of cytopathic effect, and this will also be further shown on some examples. In the antiviral studies, it is also necessary to evaluate the virus quantity. Well, in this case, we may focus either on the infectivity or actual amount of viral particles based on their genetic material. So we can do a lot of different chemagglutination tests, also plaque assays, which are easy, and they are utilized in many different virological laboratories. However, in our laboratory, we are focusing on endpoint dilution assays, which are a little bit more complicated and time-consuming, but they produce, in our opinion, better results. And on molecular methods like real-time PCR to evaluate the virus concentration. The endpoint dilution assay allows for a virus assessment of virus infectivity, whereas the real-time PCR will give us the viral load, meaning the amount of viral particles within the sample. Before we start doing anything with the viruses, we have to evaluate the toxicity of the compounds using a simple microculture tetrazolium assay, which was also mentioned a couple of times during this conference. So we evaluate the toxicity. We are also using some cancer cell lines to assess the anti-cancer potential. And of course, we are using a normal cell line like viral cell line to assess the non-cytotoxic concentrations that will be further used in antiviral studies. So we read the results from those dose response curves. Here you have the evaluation of CC50, although in case of antiviral studies, we are looking for non-cytotoxic concentrations, obviously. When we have those non-cytotoxic concentrations evaluated, we can attempt the actual assessment of antiviral activity. Very often it is based on the influence of the substance on virus-infected cells, expressed as the ability of the virus to cause the cytopathic effect. So in short, if we infect the viral cell line, which is here shown as the viral control with the virus, we expect to observe the cytopathic effect. So here we have the herpes virus type 1 induced cytopathic effect. And then we have infected cell lines treated with different kinds of extracts. These extracts were from Geranium peronaicum. This study was done with my best friend Gökhan Zengin, who is also present today with us during this conference. And as you can see here, decreasing concentrations of the methanolic extract from this geranium were used, and when we have the concentration of 250 or 200 micrograms per milliliter, the cytopathic effect was inhibited, whereas when we have used lower concentrations like 150 or 100, we could see the development of cytopathic effect. So also we could observe the dose-response relationship of this methanolic extract from this particular plant species. We also had hexane, ethyl acetate, and aqueous extracts, which were actually not active in this assay. But this is only the beginning of our work, because then we have to perform the titration assay. So we are obtaining tenfold dilutions of the samples collected from previous experiment, and then we are adding them to viral cells and observe what is the infectivity of the virus in those specific samples. And once again, we have a confirmation here that the methanolic extracts at 250 and 200 micrograms per milliliter induced significant inhibition of the viral infectious load by more than 4 log, which is regarded to be a significant antiviral activity. This type of assay measures the infectivity. However, we can also test the amount of genetic material of the virus, sometimes by using very simple approach like a normal PCR with electrophoresis, like here, but this is only a qualitative analysis, and unfortunately it also has a lower sensitivity. That is why usually we are focusing on real-time PCR, just like in this example. So here we have real-time PCR analysis of the viral load in another experiment we've done with my friend Gökhan and also with Professor Sieniawska, who had a lecture yesterday. And this was done on Spathodea campanulata extracts, which also showed us in infectivity experiments a very high inhibition of the herpes virus. However, when we've tested the viral load here, the results were a little bit different. In this assay, we've been using a calibration curve prepared using a gene-proof PCR kit, which is actually a test for in vitro diagnostics of herpes virus infection, which allowed us to obtain a full quantitative analysis, and the results were expressed as the copies of the virus in our sample per milliliter. And in the table, we've got the log out of this value. And we could observe some inhibition because at larger concentration of 500 micrograms per milliliter, the reduction was almost 1.5 log. However, the reduction of infectivity was 5 log. So there was a discrepancy between those results, which made us think what is actually responsible for the observed activity. My initial idea was that the replication of herpes virus is being inhibited at a very late stage of the viral replication cycle, somewhere around the viral synthesis of the viral proteins or maybe maturation of the virus. In order to confirm this, we've done another analysis. This was not done by me, this was done by the Turkish team, who actually did some molecular docking analysis against 3 viral enzymes. So we have selected thymidine kinase, DNA polymerase, and protease. We have tested the activity and found a remarkable result that these molecules were the inhibitors of viral protease. And now everything came together because this corresponded with the results of previous experiments. The reason why here we had a difference in inhibition was because the viral replication was actually inhibited here during the maturation, not earlier during the nucleic acid synthesis. So this was a very interesting observation. So this was just to show you how we are doing this kind of research. And now I will talk about certain problems which may be associated with performing in vitro assays, not only in virology, but similar problems you can find in many other assays you may run. First of all, the complexity of natural products. Those are mixtures of many different dozens, maybe even hundreds of molecules which may interact one with the other, which may sometimes be useful because of different kinds of synergism. However, there may also be antagonism, and what is more, some of those molecules may be toxic to the cell, masking the ability of other compounds to influence the viral replication cycle. So if those molecules are toxic, we have to use very low concentrations of the plant extracts, which actually makes it impossible to see potential antiviral activity of certain molecules. I think all the people who work with natural products and in vitro assays share one similar problem, which is the solubility. Many extracts, they don't want to dissolve in organic compounds. Not all organic compounds, solvents may be used in vitro studies. So often the solvent of choice is dimethyl sulfoxide, which is okay, unless you are thinking about the toxicity. Here you have to watch out for the proper concentration of the DMSO. However, if you keep this in mind, it is okay to be used. However, often we have a situation where the extract dissolves in DMSO nicely. However, when you add them later on to the liquid media, which is water-based, then once again you have the solubility issues because certain molecules, they do not want to solubilize in liquid media. Sometimes there is a necessity of isolation of pure compounds. Of course, for chemical purposes like the evaluation of the structure, usually the amount of compound is relatively small. But if you want to test those molecules in vitro, you need a definitely larger quantities of isolated pure compounds, which is sometimes a huge problem. Another thing to consider is the influence of the different molecules on the cell cycle of the cells you are using for viral replication. If there is any influence on the biochemistry of the cell, this might not be seen during cytotoxicity studies, but this may influence the replication cycle of the virus, which is not what we are actually looking for. We want the substance to block the replication by influencing the viral enzymes, not by influencing the cell. Another issue I would like to mention about is associated with volatiles. So we've been doing a study with Professor Sieniawska concerning on microemulsions of essential oils, and we compared the microemulsions with the essential oils dissolved in dimethyl sulfoxide. We've tested the cytotoxicity and so on, but we've also did a study on how much of those compounds is being lost during the incubation period. Because in biological studies, you often incubate the samples in 37 degrees Celsius. Obviously, if you have a volatile, it will evaporate. So we wanted to know how fast those molecules are actually evaporating. In this small example, you can see, in case of eucalyptol, that after 24 hours, it is completely gone from our mixture, which is a problem because when we are doing antiviral studies, usually they take between 48 to 72 hours of incubation period in 37 degrees Celsius. If you are losing those molecules after only a day, then what's the point of doing this kind of research? So this is also a very important thing to consider, the volatile nature of certain molecules. Of course, another problem is the influence of the incubation on the compounds themselves. They may change, they may undergo chemical modifications, they may be metabolized by the cells, and different kinds of derivatives are being produced, but that's a completely different story. The thing as I was also mentioning before, so the influence on cell cycle. We have numerous examples of natural products interfering with the biochemistry of the cell, influencing a lot of different metabolic pathways. And this also has to be considered when developing new antivirals. We don't want these molecules to be interfering with the cell cycle because we don't want those products to be harmful to patients, obviously. So that's also an important issue. Also, not always the thing we are observing in vitro can be repeated and observed once more in vivo. So there is a problem in implementation, especially of the in silico studies, then further into in vitro and in vivo assays. In virology, we may also have problems with controls. Obtaining a negative control is very easy because there are a lot of compounds which do not have antiviral activity. However, obtaining a positive control, so substances with antiviral activity, may be problematic. In case of herpes virus, it's easy because we have acyclovir and many other antivirals, but we do have viruses like Coxsackie viruses where we do not have any available approved kind of treatment. So also it is difficult to show in research or further on in a paper a substance which will be a positive control. Of course, some people use ribavirin, but ribavirin has limited activity, and thus it's not a perfect positive control for Coxsackie virus, for example. Okay. Another thing I've also seen lately are some problems with ethical perspective towards animals. I've reviewed a couple of papers recently, which, in my opinion, were hugely controversial as far as animal testing is concerned. So basically, keep this in mind always with proper respect to ethical approach to science is necessary. I would also like to encourage everyone to publish their negative results because they are also very important, because they are showing other people which direction is not appropriate or what kind of activities are not, or observations are not needed to be followed anymore because they've shown negative results. So in my opinion, also showing these negative results is extremely important. So this would be end of my talk. So thank you very much for your attention, and I'm eager to hear and participate in your own presentations today. Once again, thank you very much.
Thanks so much, Łukasz, for the very interesting lecture. Really, really very huge work. We have just a time for a question from Professor Łukasz Świątek. I see the hand of Ricardo Diego. Please, Ricardo.
Good morning to all, good afternoon too, at the heroic time. Congratulations, Dr. Łukasz Świątek, for your presentation. Great. And my question is about the use of some types of formulations, as for example, nano emulsions, since they provide more dispersibility and also penetration at the organism, at the sites of action. It could be used to improve the access of the compounds to this action, to these antiviral actions. Thank you.
This is a very interesting question. And actually, we've did some research using microemulsions. I've shown some of these results, but I did not show the cytotoxicity part. The problem in this research was that the microemulsion itself was toxic. So actually, the result of cytotoxicity obtained for different microemulsions for different essential oils containing microemulsions were very similar. And when we've tested the microemulsion, the base itself, it was toxic. So this is a problem. And also, when during the studies of the release of those volatiles, we've seen very similar results for microemulsion and for the essential oil dissolved in dimethyl sulfoxide. However, we are currently also testing some nano formulations using essential oils, and these were actually less cytotoxic, but they were actually quite non-cytotoxic because we've tested concentrations up to 2,000 micrograms per milliliter. However, we did not perform any experiments on the release of those volatiles yet. We probably will do it in the future, but now we just did not have time to test it. But that's a very interesting question. And that it would be also very nice to observe how those nanoemulsions influence the activity in case of antiviral studies. This would also be very nice. However, one thing I've noticed about nanoemulsions is that sometimes during the incubation, maybe some of the components of those nanoemulsions may be interfering with biological assays. That's a thing that I've recently observed. And sometimes those, because in emulsions you are using different fatty acids, for example, for obtaining those nanoemulsions, and they may stick to plastic surfaces in Malta whale plates. That's another thing I've noticed.
Oh, thank you so much for your response and clarification. It'll be a pleasure. Thanks.